Good evening, students. Is the slide visible? Hello? Is the slide visible? Anybody can tell? Am I audible?
Yes, ma'am.
Okay. So today we'll be dealing with the topic that is cytoplasmic inheritance. Actually, before last class, we have I have not taken the class since almost 15 days. That's why suddenly I took a class on breeding. But for now, we'll be continuing with the genetics and around after some classes, we'll be continuing the breeding class. Okay. So please mute yourself. Okay. So let's begin with today's class. Today we'll be talking about three topics: that is cytoplasmic inheritance, then genetic code, and next topic is central dogma. Okay. Cytoplasmic inheritance is a little bit bigger one, and the next two topics are small only. Okay. So let's begin with it. Okay. So what is cytoplasmic inheritance? As we all know that means till now we have studied that whatever inheritance and whatever things are inherited from one generation to other, that is because of nuclear thing. Okay, but whenever some cases are like whenever you see there is no role of the nuclear gene, in that case we may tell that there is the effect of, that is the effect of cytoplasmic inheritance. Okay, so there are certain sort of conditions where you can call it a cytoplasmic inheritance. And see, the first case is like when the classical segregation, you cannot see the classical segregation pattern, that is the Mendelian pattern of segregation. When you cannot see that, that condition might be because of inheritance. When this, okay, then whenever your trait is showing, whenever your trait fails to show any, means it cannot be grouped into any linkage groups. Linkage groups mean each of the gene, whatever nuclear, whatever chromosomes any organism is having, it is having on that genes are present on each chromosome. So let's say if the 2n is equal to 20, as in case of maize, so it will have 10 chromosomes are repeated, means 10 chromosome pairs are present. Okay, so on that 10 chromosomes, the whatever genes are present on one chromosome, they have the tendency to inherit together. That condition, in that condition we call all the genes present on a particular chromosome as belonging to one linkage group. Okay, so that is the case. So whenever you find a trait which you cannot link or associate with any of the linkage groups, in that case you may tell that there is no role of nuclear genes. That might be a role of cytoplasmic genes. Okay, and when you see reciprocal differences in reciprocal crosses, in that case also you can tell that that is basically because of your extra chromosomal inheritance. So that is all about the means what we define the cytoplasmic inheritance. So what who cause the cytoplasmic inheritance? Apart from nucleus, whichever organelle your your genetic material kind of thing are present, DNA or DNA is present. So that organelle might have some role in cytoplasmic inheritance. Other than that, if some sort of virus has invaded some organism like bacteria or human being, in that case also they tend to express they used to release some particles, like we will see in case of your pocky mutant and all those mutant in case of mitochondria, and some virus invasion in case of your bacteria. Yeah, they used to form some particles which cause some sort of inheritance, and that is also transmitted from cytoplasm to next progeny cytoplasm. Okay, so in that case also we used to call it as cytoplasmic inheritance. Okay, so there are various examples of cytoplasmic inheritance mentioned in this point. I want to add a few things like okay, that will be I'll be discussing later on. Okay, so let's go through the examples of cytoplasmic inheritance. From these examples, now, they may ask you in the ask you in the exams, like they will put the trait like like snail coiling is because of what, is the indication of what. So you need to write it like cytoplasmic inheritance. Okay, or if specifically they will be telling like maternal effect or something, then it is maternal effect, it is governed by maternal effect. I'll be telling you maternal effect is a part of cytoplasmic inheritance. Okay, okay, so let's go through in brief. That is, first one is plastid inheritance in Mirabilis jalapai. You can see that is a type of cytoplasmic inheritance, and most of the plastids which are inherited, that is not only in Mirabilis jalapai, in other crops also, that will be plastidial inheritance. Okay, that is by that is by chloroplast. Okay, next is shell coiling in case of snail. In garden snail, you will be seeing two types of coiling, like left-handed coiling or right-handed coiling. Whatever coiling the snail will be acquiring, that will depend on the cytoplasmic inheritance. Okay, so next is which are present in paramecium, they will not allow the other strain to grow. And the strain, based on this presence of kappa particle, there are two types of strains, like killer strain and sensitive strain. Sensitive strain, when they come in contact with the killer strain, they used to acquire these particles and they used to die. Okay, so the inheritance of these kappa particles are also cytoplasmic inheritance governed. Then this is the famous example, that is cytoplasmic male sterility in case not only in maize. For example sake, I have not written in maize, but a cytoplasmic male sterility always and always is a cytoplasmic, a term already showing you. But the thing to understand here is that cytoplasmic male sterility not governed by, there is nothing to do with your chloroplast. It is totally mitochondrial in origin. Okay, you need to remember that. Then sigma viruses in Drosophila, milk factor in mice. These two are not that much important. But these two, Pocky mutant in Neurospora and Peptide mutant in yeast, these are two famous examples. Okay. Okay. Then let's go to the next one. Okay. So what is seen here is they have divided the total hereditary material into two parts. First one is plasmon, that is the all the hereditary material present in the cytoplasm is called plasmon. Okay. Then second thing is genome, that is the all the hereditary material that is present in the nucleus, that we call as genome. Okay, so plasmon and genomes are two terms. There will be some more terms like present over here. Like first one is plastid. The in case of the cytoplasmic inheritance, based on which organelle is involved in that particular inheritance pattern, they will be talking about, they have given the terms. Okay, and whichever organelle is having the particular DNA, based on the location of DNA in that organelle, we'll be calling that DNA as either chondriosome, chondrion, or plastone, like that. So here they have given that plastogenes. This is also called is that plastone. Okay. Then next one is chondrion. Chondriogene that is found in case of mitochondria. It is also called as chondriome. Sum total of the genes found in mitochondria is called chondriome. That is I have mentioned somewhere in the later slides. These two things, they centriogenes and kinetogenes, they have reported in some bacteria, but their identity and role in case of cytoplasmic inheritance somewhat ambiguous. But these two, plastid and mitochondria, are very much prominent in case of a cytoplasmic inheritance. Apart from this, you can see the famous term that is plasmid. You must be aware of it, right? Plasmid is, what is the plasmid? Anybody can tell me what you understand from the term plasmid? Yes. Yes, you can mention the term. What do you understand? Yes, it is self-replicating in nature. Anything else? Okay, plasmid is nothing but it is a circular thing of DNA. Yes, it is circular. And it is a circular, small circular DNA that is double stranded, mostly double stranded in nature. And that is not your genomic DNA, that is epigenomic DNA. Find the nucleoid, that is a circular genomic material itself. Apart from that main genomic material, you will find various little small circular double stranded DNA apart from that, that are physically separated from your chromosomal DNA, and they have the property like they will be harboring various genes, and they will be replicating independent of your chromosomal DNA. And they are present in various numbers like one or two. They may be present in one or two, or they may be present in multiple number based on what they do, what action they have, and what they do for the cell. And basically they used to have some good, means they used to have some beneficial roles for the survival of the bacteria or the organism carrying it. So they used to propagate, and they used to carry this particular plasmids from generation to generation. They may harbor sometimes some antibiotic resistance gene, like the famous example of F plasmid, that is for, that is also called as sex plasmid. That because of that the conjugation becomes possible, and two types of variability is created in case of bacteria. And other types of plasmid like R plasmid and other plasmids are also there. And we have explicitly used the plasmid in genetic engineering, that will be dealing in the biotechnology section. Okay, so and this plasmid is also part of cytoplasmic inheritance, because it is in cytoplasm. Whatever thing is present apart from nucleus, those things will be coming under cytoplasmic inheritance or extra chromosomal inheritance. Okay. Okay. Then I have mentioned some points to ponder. Like we'll be going through this slide later on also, but still I'll be going a little bit through it, because this slide is very important from your objective type point of view. Whatever thing I have explained here, if you don't, if you cannot understand for the time being also, just stay tuned. We will be getting to all the points somewhere in the slides. And at the end of the lecture also, I'll be going through this slide again, so that you'll be understanding it better. So cytoplasmic inheritance is also called as, sorry, cytoplasmic genome is also called as plasmon, organelle DNA, extranuclear DNA, non-nuclear DNA. Anything can be asked in your exams. Okay, so you need to remember that. And these are the gene controlling the cytoplasmic inheritance are present in the nucleus, outside the nucleus, that is in the cytoplasm, and they are known as plasma gene. Otherwise, somebody will tell you the other names of your plasma gene. So it is also called as cytoplasmic gene or extra chromosomal gene. Based on this, they used to call this inheritance as cytoplasmic inheritance, extra chromosomal inheritance, or maternal inheritance. Why they are called as maternal inheritance? Because they are transmitted from the maternal parent to the progeny, and again from the maternal parent to next progeny. Okay, and why that is happening, we'll be discussing later on. Okay, just remember all these points. DNA, what it is called. All the genes present in the all the genes that is present in a plastid is called plastome, and whichever gene is present in case of mitochondria, the chondrium term is for that. I already told you, right? This is for mitochondria. Sorry, during typing some mistake is there, so just bear with me. And here the sentence is given for that is for plastome, that is a plastid, and chondrium means the all the genes that is present in mitochondria. Okay. Okay. Then plastid inheritance was first discovered and characterized by Carl Correns. This is important. And he has studied this in 4 o'clock plant. This is very famous plant for experimental material. And the many scientists have worked on this plant, that is Mirabilis jalapa. He has studied the leaf pattern, leaf variegation pattern. We'll be seeing that in detail. And what is leaf variegation pattern means presence of what kind of leaf coloration it is giving. And on that leaf coloration, what is the impact of your cytoplastid, that is your various kinds of plastid like chloroplast, chromoplast, leucoplast, everything that he has studied. Okay. And total gene that is present in the cytoplasm is called as plastome. And so here I have mentioned that sentence which I missed here. Okay. Then this is the point, this is extra information just for your knowledge sake. The lower plant that is liver root, that is Markensia. You would have studied this so many times in your 12th or 11th. And this plant is very important in that it was the first plant for which the mitochondrial genome has been sequenced. Okay. This is for your information sake. And this is also one piece of information, no need to bother about that. Okay. So coming to the history of the organelle genome, that is when and how we proceeded with the organelle inheritance and all those things. So in the early 1950s, Sanger was a scientist. Sanger, you would be will be studying this particular scientist name in so many things. Like he has a big role. He has a big role in case of your genome sequencing. Okay. Sanger sequencing you might have heard somewhere. Sanger was the first person who has given the uh, DNA sequencing mechanism. It is just for information. Okay, we'll proceed with the history. So he and his colleagues first suggested that there might be some role of your cytoplasm in case of inheritance. So the first evidence was given by Correns in case of Mirabilis jalapa in 1908, and simultaneously bore another scientist. He has given the evidence in case of Pelargonium zoner. Okay, and Jenkins was the first to describe the Ioja variegation in case of maize. From Ioja variegation, it is like in case of your maize leaf, it is like this. Let's say it is a maize leaf. You can see pattern stripes. We'll be seeing the diagram later on. Like this stripe will be green, this stripe will be white, this stripe will be green. Like that pattern, that is called Ioja pattern, leaf variegation. And from here, how they used to ask the question, scientist is important, but the crop is much important. Like Ioja variation is seen in which crop, they used to ask. Okay, that is seen in maize. And the next thing is Rhodes. Rhodes has listed the CMS in maize. Here the scientist is very, very important, like because it is cytoplasmic male sterility. So anything related to male sterility, mostly they used to ask in the exams. Okay. And this scientist, that is Sonborn, uh, you can remember this, like he has studied the Correns is and Rhodes has given the term that is CMS in case of maize. Okay, next thing. Next is organelle okay, organelle DNA. So what is organelle DNA? It is also called as minor DNA, right? It is also called quaternary DNA. It is of two types, mitochondrial and chloroplast type. And the features of organelle DNA, it is very, 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 very important. Hello, students. Sorry, there is a rain in my area and I am facing the internet connection continuously. Sorry for that. Are the screens visible now and slides visible now? Yes, anybody can tell me? Okay. So, so organelle and DNA, what I was telling here is important feature is both mitochondria and chloroplast. They are semi-autonomous. Okay. The term here is important. That is semi-autonomous. Why they're called so semi-autonomous? Because autonomy means in the sense that whether they can survive on their own, means won't depend on nuclear thing or anything, but they are not completely autonomous. They have to depend on the nuclear genome for some sort of proteins or other thing, but they can produce some of the protein which is necessary for their survival, and because of that reason they are called as semi autonomous. Okay. And they are liable to mutation, like various kinds of mutation occur here, and whatever mutation will occur here, since it is present in only means one allele is there, so if the condition is of recessive case also, it will be expressed here. Okay. And they are expressed and inherited separately from nucleogenes, I have already told you. And they are transcribed and translated within the organelles, but for some of the proteins, let's say for example your proteins associated with electron transport chain, where they will be found, in which organelle electron transport chain is found? Anyone tell me? Yes, anybody? Please respond. Yes, it is in mitochondria. So here you would have known that 4 or 5 complexes are there and several proteins and molecules are involved. So for that, whatever proteins are required, whatever enzymes are required, partially they are synthesized by mitochondria. And let's say for a protein, there are several subunits are there. So some subunits will be synthesized by the your mitochondria itself, and some will be processed or produced by the nuclear genes, and they will be transferred to your mitochondria. It is like that. In that sense, it is semi-autonomous, and it will show uniparental inheritance because they are generally inherited from the female parent. Why they are inherited from the female parent? Because as you see in all the cases, like in humans also, in plants also, you see the egg cell is much more bigger than your pollen, or the very big and this is very small. Means pollens normally do not carry your pollens do not carry your cytoplasm. Very little cytoplasm is there in case of a pollen, and that is just for its survival. And pollen do not donate almost any cytoplasm to your at the time of fertilization. It only donates your nucleus. Okay. So whatever cytoplasm is present in case of egg, that is from that only the zygote will form. So the zygote will carry the cytoplasm solely from your mother parent. Okay. Means maternal parent. So the female parent cytoplasm is itself is coming to your progeny, and that's why we are calling it as and the all the organelles and whatever thing like cytoplasm, chloroplast or mitochondria, they are present in the cytoplasm only. No, so because of that region, all the organelles along with the cytoplasm is coming. That's why whatever organelles are present, whatever organellar inheritance or whatever pattern is there, in case of your maternal parent, that thing is carried forward to your progeny. So that's why it is called like uniparental inheritance, because paternal parent or the male parent has no role in that particular inheritance. Okay. Because it is not contributing any sort of cytoplasm to the progeny. So it is, that's why it is called as uniparental, and generally it is from female. And reciprocal differences are observed here. Here why reciprocal differences will be observed? Because if you just intercross, reciprocal cross means, anybody can tell me what is reciprocal cross? Hello? No? Okay. So if you will just cross A and B, let's say two parents, that is A and B. A is the in the cross, basically we used to write the female parent first. It is the female parent, it is the male parent. Yes, it is exchange of parent. Exchange of parent means just the male parent will be okay. Let me explain. So in the first cross, that is A is used as your male, A is used as female, and B is used as male. So the progeny will be carrying the cytoplasm of solely A, isn't it? Okay. Then if you just rotate the cross, like your in the reciprocal cross is, in this cross is called as direct cross, which will plan. But if you just alter the parent, like B will become the female parent, A will become the male parent, like B has become female and A has become male. Okay. So this condition is called as reciprocal cross, and in this condition, what the in this progeny of this particular cross, what cytoplasmic genes will go to the progeny? Here, whatever cytoplasmic genes will be so that's why they're calling that reciprocal differences are observed. So this is very important. And how it is observed, I think it's clear. Then it will show maternal effect. Maternal effect I will be discussing you in the next two or three slides explicitly. Let me tell you the definition what is maternal effect is. Maternal effect means whenever the particular trait is inherited or governed by the genotype of the maternal parent, not on the progeny's own genotype. Genotype of the maternal parent is important. Nuclear genotype. Okay. Nuclear genotype of the maternal parent is important. In that case, it is called as maternal effect. Okay. And maternal effect and maternal inheritance. Maternal effect is a subset. You can call it as a subset of maternal inheritance. It is not solely. But maternal inheritance is the total cytoplasmic inheritance. Here there is no role of your cytoplasmic. Here there is no role of nuclear gene. Here the inheritance, whatever is happening, that is because of the all the organelles that are present in. Let me clear this. It has become very dirty. Okay. So whatever inheritance is going on, that is because of the genes that are present in case of your mitochondrial chloroplast, not the nuclear gene. But the maternal effect is something different. You will see, what you will see here is the effect, the progeny's phenotype. Progeny, what particular trait, let's say something else, like coiling of snail, that is the example of maternal effect. We'll see that in detail. So whether the progeny will be left-handed coiling or right-handed coiling, that will depend on what was the genotype of the mother. Okay, that is what called as maternal effect. Okay. Then the mapping is difficult because they are tend to harbor many sort of mutations and all those things. That's why mapping is a bit difficult. And we have not explored much about this. That's why also mapping, we may call it bit difficult. Then they do not show in sort of segregation in F2 and Mendelian inheritance. They show non-Mendelian inheritance basically, because they are not they are uniparentally inherited, so they cannot show any sort of Mendelian inheritance. Mendelian inheritance is what we studied in the earlier lectures, like they have to show 3:1 segregation for one gene, then 9:3:1 for second two gene. Like that, there is no segregation pattern. And why they will not segregate? Because there is nothing like means meiosis or recombination or anything. Here they will show recombination among each other, but they are not segregating in any case. Okay. Sometimes we will see some sort of mitochondrial recombination events, but they will not segregate. Then they are self-perpetuating hereditary particles. Okay. And self-perpetuating in the sense that mitochondria can replicate itself. Chloroplast can make another chloroplast from itself. In that way, they are self-perpetuating. Excuse me. And they will show the somatic this chloroplast having this is the total chloroplast. This is not a chloroplast, this is leucoplast, and this is chloroplast. This is, let's say this is green in this is some color, blue in color. Okay. So when the cell is dividing, somewhat like this, there is no particular pattern that your organelles will be separated half to both the progenies. They will be distributed randomly. So whatever the progeny cell will be having, nobody can tell that exactly, okay, here 3 copies were there, so same 3 copies will be there here also. They might be there or might not be there. And from these 3 copies, which copy will go to which parent, it is not decided. So randomly, let's say to one of the progeny cell, it carried only the chloroplast and the leucoplast, but the chromoplast is not there. Here the chromoplast is there, okay, that is the blue color. So whichever cell will be growing from this, they may express the color, if they are exposed to light, they may express the color like green color, okay, green color. And if they are not exposed to light, they may be albino, means white in color in case of plant, okay. And in this particular cell, because it does not have any sort of your chloroplast, and that is because it will go for the blue coloration in case of leaf. You would have seen the ornamental plants in your horticulture floriculture lecture or floriculture course. There are several plants we are using ornamental purpose. They have various kinds of colorations, like the plant, I'm just forgetting its name. We used to call it as I think Corium variegatum. I just forgot. Sorry. So so we used to call in that particular plant has various patterns in the leaves and spots, different coloring spots are there. So wherever you will be finding that particular thing, that is because of this particular somatic segregation. Okay, and because of in that particular region of the cell, that particular region of the tissue, the cells have expressed that particular kind of plastids only. That's why this coloring pattern is coming. Okay, moving to next, that is what is the difference between, I told you, no, like your Mendelian and non-Mendelian inheritance. So what is the difference between them? Just to I'll be discussing the striking features, some of them. So Mendelian inheritance is governed by the nuclear genes. Here non-Mendelian is governed by the plasma genes, and here they will show the distinct segregation pattern, that is Mendelian or non-Mendelian, whatever it is. But here no distinct segregation pattern is there. Reciprocal differences not observed, but here reciprocal differences you can see. And they do not show any maternal effect, but here they will show maternal effect. Okay, and but it is not true, means if you just see maternal effect, genes are themselves uh, showing the Mendelian inheritance, because they are governed by the nuclear gene, but they are governed by the nuclear gene of the parent, maternal parent, not their own genotype. In that case, you may call it as non-Mendelian in the progeny, but it is the maternal genotype, whatever present in the mother, that is expressing, and that is segregating in the Mendelian fashion only. I'll be showing you how it is happening. Don't confuse in maternal effect. It is a little bit difficult to understand, but I'll be telling you later on. Okay. Then genes can be mapped on the chromosome easily. I told you they can be mapped to any particular linkage group, but here the mapping of genes is not possible. So what is the difference between them? Same like the carrion also observed by some lower organisms, but mostly that is a generalized rule that your pollen or your male parent will not contribute any cytoplasm. Okay, in some cases there are some exceptions. The next is that you see in the diagram he present over here, this is the nucleus, and this is your mitochondria, and this is the chloroplast. I told you, like the fertilization process, the chloroplast from here, that is a female parent, and the mitochondria is coming. There is no chloroplast or mitochondria coming from male parent. That was illustrated here. Okay, so let's just get some more information regarding our mitochondrial DNA and chloroplast DNA, since those two are important aspects in case of your cytoplasmic inheritance. So this will be either mitochondrial DNA can be either circular, but in some cases, in some fungi, they have observed that it to be linear. So it is an exceptional thing. Okay, if somebody asks you the shape of the mitochondrial DNA, it is exception. Sorry, spelling is mistake. Okay, so I can write it. Okay, so it is generally circular, but rarely it is linear. Size will be means mitochondrial DNA size. In case of plants, I'll be telling you the plant mitochondrial DNA size is bigger than your plant chloroplast DNA size, chloroplast of plant, and both of them are bigger than mitochondrial DNA size of animals. Okay, so in animals, smaller size of mitochondrial DNA or mitochondrial genome is found. Okay. Plant mitochondria. Another thing is that in many of the books you will find that mitochondria or chloroplast genomes are intronless, but in case of plant mitochondria, that is the exception that plant mitochondrial DNA carries some sort of introns or non-coding regions. Okay. Then next is chloroplast DNA. That is but in the exam, if somebody is asking overall, means not specifically about mitochondrial DNA. So like in the chloroplast or in the cytoplasmic genomes, intron is there or not, because they are told to be derived from your eukaryotic genome. So they should not carry any sort of introns, but because of the evolutionary aspects, they have acquired some sort of non-coding genes. But in generalized condition, if somebody is asking, it will not have any introns, because most of the organisms do not have intron in mitochondrial DNA, but plant is having. Okay. Then chloroplast DNA is double stranded, circular, super coiled, and it will not have any proteins like histones or anything. And size, I told you, like it will be somewhat around 85, means there is a big range over here. Then mm, no, this piece of information is not correct, I guess. I'll be clarifying you this, but somewhere in book I have studied that what I told you, like mitochondrial genome will be bigger and fetus contains about 10 to 60 copies. Whatever DNA are present, they are circular DNAs, and they will not be present in one copy. They'll be present in several copies. In mitochondrial DNA also and in chloroplast DNA also, both. They will be present in several copies. Then what is the common between mitochondria and chloroplast? Somebody has texted something. No. Wherever it is important, I'm telling you, no. Just like for your information sake, I have given all this thing. Wherever I had mentioned, like, because while you be reading for exams, no, you'll be covering only the important points. No need to go through all these things, all the slides I have given. But the thing is that for the sake of your understanding and for ease of your memory, that you can just frame a picture in your mind so that you will remember all the thing. Okay. Or else you will be confusing somewhere if you just only mug up the things. That is also possible that okay, but you may confuse. That's why I have given some of the facts. But wherever it is important, I'm telling you already, wherever it is important, you, for the examination point of view, you focus on that point only. Wherever I'm not telling anything, means like just uh, going through the slide or telling you something, some facts, it is not that much important, but it is for your understanding. Okay. Then next thing is here. Okay. Forget about the to both side things. Here the same thing is that mitochondria and chloroplast both have the provides a cell-based energy. So here the important thing is that in mitochondria, via respiration, via respiration, your energy will be produced, and in chloroplast, by the water and substrate level water phosphorylation, all those things are happening, and in the end here also some energy is getting generated. Both are double membrane structure, both have their DNA and RNA, and they have similar enzymes and coenzymes, because they both of them are producing energy. That's why. Okay. Then this is important, like endosymbiotic theory. What it tells you actually, endosymbiotic So how your mitochondria and chloroplasts have originated, that's it. This is the only point you need to remember from this. Hello? Am I audible? Hello? Hello? I think I'm audible. Okay. So let's continue with that. Okay. So what they're telling here is, just let me change my internet connection once. Okay, so here what is happening is, your mitochondria was originally an heterokaryotic prokaryote, means it was not having any photosynthetic pigments with it. It's then called by your cell, and it became your mitochondria. And another photosynthetic prokaryote that has the means that has the photosynthetic pigments, which was able to photosynthesize, that was engulfed by the cell, and it became your chloroplast. Okay, that is a theory that was called as endosymbiotic theory, means there is a and lattice of mitochondria and chloroplast, they are similar to your uh, prokaryotic nature. Okay, prokaryotic bacteria or whatever it is, no? Prokaryotic organisms. Then significance is, they have the role in case of your transmission of a character, leave it. All these things are not that much important. And cytoplasm male stilate, it has the importance, you need to remember organelle and DNA, that is your mitochondria. Okay, then these three examples I already told you, like cytoplasmic instability, pockiness in neurospore and petite is, these three things are governed by mitochondrial inheritance. Okay, that you need to remember. And rest of the things are not that much important. I'm skipping it. Mm, okay. Kappa particle, I told you, like in Sonborn was given this inheritance. Two strains, like killer strains and sensitive strain. And they have governed by the genes, like capital KK and small kk. The killer cell secretes the toxic substance called paramycin. They might ask you the chemical name like paramycin, which kills the sensitive strain. So whenever these two particular strains are together, one in the beside another, the killer strain used to kill the sensitive strain, and only killer cells survive. And by conjugation event, they used to transfer these particular things to one another, and further the killer strain, sensitive strain becomes killer strain, something like that. So from here we need to only remember the kappa particle. There is a particle, it is not any organelle or something, nothing like that. It is simply a particle, toxic particle, which can secrete a substance that is paramycin, and it was discovered by Sonborn. That's it. Then leaf variegation is, it is important. Few important points only I'll be discussing here. I already told you this is those were given by Carl Correns. And variegation means the white and yellow spots, white, yellow, or green spots, whichever you see in case of leaves, that is called your variegation. And in case of Mirabilis jalapa, the photograph is here, you see. In case of, this is the plant of Mirabilis jalapa, this is a real picture. You see here, this is the green thing. This is here, you can see some yellow thing here, and you see the pure white thing present over here. Okay, so what they're calling is as, this is the variegation pattern. That is, green one is having chloroplast, pale green one is having leucoplast, and variegated one is having all the three types of plastid. So based on which region, in this particular region it is only white, means here only leucoplast is present. In this particular leaf, you see some portions are yellowish and some portions are, the portion is white in color. So this is having some the chromoplast, and this portion is having some leucoplast. Okay, and whatever is fully green, this particular leaf is green, so it will be having only chloroplast, like that. So the phenotype of the progeny resembles the female parent, and what is happening is, from a particular branch, whatever in this particular branch you see here, only the bird was, whatever the leaf bird, whatever leaf bird originate from which the branch is originated, whatever chloroplast it will be having, whatever plastid it will be having, based on that your whole branch coloration will depend. Here it is completely green, so we may call it that, like the bird will be having, the tissue is green and only your chloroplast were present. But you see here in this particular region, it is quite means mixed kind of thing are there. That's the use of particular means variegated pattern is observed over here. In this particular region, there is no chloroplast. So whatever branch has come upon on this particular part, this particular inheritance is called variation in Mirabilis jalapa. So the thing to remember over here is the scientist name Carl Correns, and here you can see the irrigation pattern. That's it. For your understanding, all these things I have given. No need to bother about them. Okay. This one also I have given for your knowledge sake. No need to bother about them. Okay. Yeah, cytoplasmic innate genes can have other roles, like they contribute to cytoplasmic DNA. We are talking about maternal inheritance, no, and cytoplasmic inheritance. So they might have role in other things like photosynthesis, respiration. We may use them as in phylogenetic studies and all those things. Don't bother about them. Next, come to the important thing is maternal effect. When the expression of the character is influenced by the genotype of the female parent, that is called maternal effect. Okay, and that is the famous example is coiling pattern in case of snail, that is Limnia peregrina. Okay, so here pattern, two types of pattern I told you, like right handed means right hand, that is lateral coiling, left handed, that is sinistral coiling. This is the right handed coiling. Sorry, this one is right handed, this one is left handed. Left handed is, you can see here, this is sinistral, this is dextral. Okay, so two types of coiling is there, and this coiling pattern depends on the maternal genotype. This is governed by the dominant gene. Two genes alleles are responsible, one is capital D and recessive, one is small d. So what, based on the genotype of the mother it is decided. Okay, So how they are decided, let's see in this diagram. Here the C, the brown ones are sinistral and white ones are dextral. For your visual understanding, they have given. And definitely they are dextrals are right-handed and sinistrals are left-handed. Okay. So what they have given is the parent. You no need to bother about this section, this particular right-handed section. You focus on the left section. Here the dextral thing is that dextral is having two particular plus symbol indicate that it is capital D, capital D. Okay, and the sinistral is small d. Okay, so the progeny, whatever what is happening over here is it will be capital D small d type. So based on the based on the Mendelian inheritance, what it should show, it should show the genotype of the capital D because it is dominant over small d. But what the thing is showing or shown over here, you see the white color is here. Why it is white color? Because it is following the maternal inheritance pattern, and it depends on the genotype of its mother. Who is the mother of this particular snail? This one. Okay, so its genotype is capital DD. So what will be the pattern observed? It is dextral. So here it is shown dextral. Okay, so here it should also show the dextral, no problem. Next you see, from here it will be segregating, like it will be capital DD. Okay, this one will be capital D small d, capital D small d, small d. Fourth thing will be there. So this one is dextral, okay, this one is dextral, okay, this one is also dextral, but this one is having the genotype small d, still it is showing your dextral pattern. Why this is observed here? Because it is having the nuclear, it is having the gene that is ss, that is present in the nucleus itself, but it is the character is governed by the female parent genotype. So who is the female parent of this particular snail? It is capital D small d. What is the what should be the expression of this particular thing? This particular thing should express like it should be dextral. That's why this though the genotype of the particular snail is small d type, it is expressing your dextral coiling. But in the next generation, you see here it is capital D, capital D. The progeny of the capital D is going to be capital D only, and based on the maternal genotype, it is dextral in nature. Here also based on the maternal genotype, here they will segregate. No, here all the because they're in heterozygous condition, they will be segregating in capital D, capital D, small d, and all those things. But based on the genotype of the female parent, it is having the genotype that is, it is having the expression that is capital D small d, which will be expressing as dextral coiling. And the sinistral one, you see here genotype is capital small d. So in the progeny, because this is the maternal parent, that's why the progeny from this particular snail is expressing the sinistral coiling based on the genotype of the maternal parent. So this particular cross I have shown for your understanding point of view. So what you need, but the only thing is the dextral or sinistral coiling seen in the snail, that is because of maternal effect, and that is governed by the nuclear gene of the maternal parent, not on its own genotype. Okay. Then coming to next thing, that is cytoplasmic male sterility. Okay. So here, just a second. Okay. So regarding the cytoplasmic male sterility, it is governed by the particular factor that is present in the cytoplasm. So it will decide whether the particular plant will be male sterile or not, that has cytoplasmic male sterility is governed by your mitochondrial inheritance, and it has been cytoplasmic male sterility has been exploited in various crops like pearl millet, sorghum, and cotton. Okay. uh, then this is the end of your topic, and from this topic as I told you at the beginning, they are important. You see, okay, so this slide is very much important. All the examples you need to remember. These terms are important. Then this slide is important. I have given the explanation for all the terms and some informations. And then history is important. Then the important features are quite important. And then endosymbiotic theory is related to the evolution of your mitochondria and chloroplast. That point is important. And cytoplasmic male sterility related to mitochondria. That is important. And leaf variegation was given by Calcorans. That was important. Next is maternal effect. It is governed by the genotype of the female parent. And it is seen in case of snail coiling. So all these things you need to remember in case of your cytoplasmic inheritance. Okay. So next thing is central dogma. This is a very small topic. Central dogma refers to the machinery. So, the process by which the instruction of DNA is being converted to functional product. That is the whole machinery of a cell by which your information is being transferred from one particular kind to another kind, so that it will be ultimately expressed. DNA is present in the cell, but DNA cannot do anything directly. Okay, so it has to be transcribed into RNA. Then RNA has to be transcribed into protein. Okay, that is the thing that should be done. Central by Crick. Yeah, central dogma concept was given by Crick. Yes. Okay. So it was proposed by Francis Crick, and Crick has another important role to play. That is, he has given the DNA double helix structure. Okay, along with Watson. And the central dogma, it is basically, it will tell you like from DNA how the thing is getting transcribed and in which direction the information should pass on. Okay. For the success of any organism, the central dogma should be very very particular, and it should be properly functioning. Then only the organism will be functioning. These two diagram, it will depict, means the clarify the things. Like here it is double stranded DNA, it will replicate itself. Okay, through transcription it will produce a single stranded RNA, where ATGC coded, whatever coded information present over here, it will carry all the information over here. The difference is that here instead of T, here U is present. Okay, instead of thymine, here uracil will be present. And this process is called as transcription. From RNA we can also produce DNA. That is seen in case of some retroviruses. This process is called as reverse transcription, and here the enzyme is important, that is called as your reverse transcriptase enzyme. Okay. Then there is another term associated with reverse transcription. It is called as teminism. Temin and Baltimore, two scientists, have given this term, given this concept of reverse transcription. Reverse transcription is also sometimes called as teminism. You just remember this term. Okay. Okay. So from this RNA, again, RNA cannot express itself. Sometimes it act as some factors, or it may modulate, or we may modulate some sort of expression using RNA, but RNA cannot express itself directly. Very rarely directly acts. It will act or it will express itself via production of protein. That is the process called translation. Okay. And again, what is happening over here? So that the same thing I have depicted, and here the protein is ultimately, here it is formed by ATGC. The ATGC coded information is getting converted into AUGC coded information, and in the protein you will be finding the linear chain of amino acid, which will be produced, and they will be encoded from triplet codes present over the RNA. Okay, three codes, three nucleotide bases will be coding for one amino acids in the protein. Okay, and those amino acid that protein sequences will be interlinked with each other to form the primary, secondary, tertiary, whatever form of proteins which are ultimately expressed in your cell to carry out the various functions. Okay, that is what the central dogma tells you. Okay, that's all about the central dogma. And next is that was a very small topic. Next is genetic code. Okay, before going for all the expression things, these things were important, that's why I discussed it. So first thing is, what is genetic code? Here the term was given by Gamow, one scientist called Gamow. I have not given that scientist name. Gammow. Gamow has given the term. They might ask the question like that. Okay, so there are various scientists who have given this genetic code, who cracked the genetic code. He is Matthew and Khurana, Hargobind Khurana, from the scientist from India, H.G. Khurana. Okay, and Matthew Matthaei. It is again, I cannot write it, and time is also not that much. So these two scientists have cracked it along with the another scientist, that is Nirenberg. Okay, and total how many genetic codes are there? Here, sorry, here total how many genetic codes are possible? 64 codes. 64 genetic codes are possible, and they are universal in nature. And these genetic codes total, why there 64, why not anything else? Because we have 4 bases, A, T, G, C, right? What is genetic code is first thing you see in this diagram. This particular thing, that is, I told you, no, here 3 bases, 3 bases will be coding for one amino acid. This triplet is called as genetic code. You see here, this triplet is called genetic code, this triplet is called genetic code, like that it will proceed. So here it will code for some amino acid, here another amino acid, here another amino acid, like that. But DNA is not coding it, it is coded by your RNA. Okay, means DNA is getting converted into RNA, RNA is getting run through the ribosome, and here the RNA codes are being recognized as whichever amino acid it will be. So that is what the genetic code is. Okay, that means that's what I'm talking about right now, their characteristic and how they will be behaving. So there are 64 codons, and 63 are sense codons, means 63 are functional codons, and 3 codons are nonsense codons. Wherever these particular 3 codons will come, you see here, these 3, this one and this one, they are nonsense codons, so stop codons, you can tell. Wherever they will come, your transcription thing will stop. Okay, and some of the codon, like this one is your start codon, that is AUG. Wherever AUG means, it is not mandatory that wherever AUG is there, it will be a start codon, but wherever starting is occurring for a translation process, AUG will be there. Okay, that is the thing. You understood it? So it will be it is always the universal start codon, and these 3 are stop codons. This is the matrix of your 64 amino acid coding codes, that is the genetic codes. Okay, and okay, and all the in the genetic code, the peculiar thing is that all of them will use the same, all of them will in total they will produce 20 amino acids, means whatever code 64 codes are there, so they will ultimately code for 60 amino acid, 20 amino acid. So the codons are 61 codons, let's say, they are synthesizing, and they'll code for only 20 amino acids. So what this directly indicate that more than one genetic code, more than one triplet codon will be coding for same amino acid. You see, these 2 codons are coding for phenylalanine, these 4 are coding for leucine, likewise. So it tells you that codons are more in number, means genetic code are more in number, and your amino acids are less in number. Okay, so what was the definition? I already told you, like they are the messenger in the RNA chain. Their codes for specific amino acid molecules, they correspond to single amino acid signal or stop signal. Okay, and yeah, This point is also important. Like each codon specifies a particular amino acid. It is true that one amino acid might be coded by several things, but one particular code, it will be indicating, or it will be recognized as a particular amino acid only. It will always, and this UUU, it will always give rise to phenylalanine, nothing else. This UCU, it will always code for serine, nothing else, like that. Okay. Then coming to the characteristic of genetic code, this is very, very important. This, all the 7 statements I have written over here. From this, they will put something in directly, like which of the following is not a characteristic of genetic code. They will put some not like the code is not degenerate, or the code is overlapping. They will remove this none from here. They may put questions from that, and they used to put the questions from here. So this is important. This slide is very important. So first thing is codon is triplet. Why? Because it is having three bases are recognized as one code. So code is triplet in nature. Then code is degenerate in nature. Degeneracy of the codon, that is another aspect. Here it tells that means more than uh, more means the incorporation of some amino acid will be influenced by more than one codon. So one amino acid will be coded by more than one codes. That's why it is called as degeneration of the codons. Okay. And they are not overlapping, like from here your codon, if the codon is starting from here in this particular thing, if your codon is starting from here, they will be continuous. They will not, it is not TGC will code for one, then CGA will code for one, nothing, not like that. If these three are already used, means they will be coding for one, then next three, then next three. So they will not be overlapping to each other. They are commonless, means there is no space between them. They will be very continuous in nature. Okay. They are unambiguous. In unambiguous in the sense that one amino acid, one codon will code for one amino acid only. So there is no ambiguity here. And the code is universal. That is this particular standard code which I have shown you, that is followed by most of the organism, except for some. In for few codons there is some changes, like the stop codon present in case of, that is the standard stop codon. They used to code something in case of animals. No need to bother about that, but codon is universal. It is followed by most of the organisms. Okay. Codon is the show the collinearity aspect. That is, they will be expressed like they will be expressed in a collinear manner. I have forgot what was the explanation for this. I'll be telling in the next class. Okay. Sorry for that. Please pardon me. Okay. So next thing is that it is a triplet code, and here you can see that here how the things is getting. Here three codes are there, and here your three codes from here AUG methionine is coming. Then from next three, this particular methionine is from this histidine is coming likewise. So this is the chain of your mRNA. From this, the encoded information is getting transformed from this genetic code, triplet code, to one amino acid. Okay. And after that, your peptide chain is there. And here one important thing, I think in mutation we would have covered. I think mutation class is over. Is the mutation class over for you? I think it's over. Okay, let me tell you again. And if there is a class called friendship mutation, so if anything is getting lost from this particular thing, either this one or two, until the case that the lost sequence or the added sequence is not a multiple of 3, means there is anything other than 3, 6, or it is not a multiple of 3. In that case, there will be the mRNA, the amino acid chain produced by the altered thing or mutated fragment will be changed. Okay. And and the initial fragment will not be same, means it will be affecting your ultimately the protein sequence. But if it is occurring in sort of 3 or 6, let's say among these two, three nucleotide bases have inserted. So this will be coded as normal as methionine, and this will be coded as normal. Only one amino acid has inserted. This whatever thing has inserted over here, it will be coding for something. So it will not change the whole and sole property of a protein, ultimate producing protein. So that is not going to hamper much. So that so the frame shift mutation will be seen if your alterations, if your mutations is not in the multiple of 3. If it is in the multiple of 3, it will not show any sort of frame shift mutation. It will show some mutation because something is inserted, but frame shift mutation will not be there. Frame shift means this is the frame and the frame has shifted. If something is changed, means the AU will not be read as AU now. AUG is lost, so AUA, then CAC. So original frame was AUGSCA, but now it is read as AUA like that. So this is the concept of frame shift mutation. Okay. And there is another thing important here, that is Wobble hypothesis. This hypothesis tells that the third base of your codon is not specific, not that is less specific. That is this thing. This thing is less specific, and whether these two only specifies which amino acid it is going to produce. Okay, so whether it is A or C or T or G, mostly it will synthesize the same, it will synthesize same amino acid. Why you see over here CA, all these things, first two bases are same, but here UCAG, whatever it is changing. So here CA are same. Whenever you, if instead of U, let's say C is coming, then also it is synthesizing the histidine. And here you see if in case of A, sometimes G will come, then also it will produce the glycine. Okay, so and here you see for CU, first two bases are same, and this last base is changing. For all the four bases UCAG, it will code for leucine only. So this hypothesis tells that whether the if the any change is occurring in the third base, it will not cause much of the difference, and it will only cause a silent mutation, that is no change in the expression, because mostly the first two bases only determine which amino acid it will be which amino acid it will be recognizing or producing. Okay, and it was given by Crick again. Okay, that was I missed to mention over here. So that's it for today's class. Okay, any doubts, students? You can ask. And another thing is that verbal base is third base in case of your codon, and codon is the sequence present over mRNA, that is codon. Okay, mRNA sequence, this is codon. Sorry, this is codon, and this is the mRNA, this is the codon, and this is recognized by tRNA. In tRNA, three bases will be there, and this is the structure of tRNA, like the clover leaf structure. This is the condition in case of your tRNA. So here you can find three amino acids which are complemented to this. Okay, so that is called as anticodon. Okay, so this anticodon and codon, they used to recognize each other during the translation process. We'll be studying this in detail in case of translation. So Wobble base, which one will be the Wobble base? The third codon of third base of your mRNA, first base of your tRNA. Third base of your mRNA means third base in case of codon, and first base in case of anticodon. Okay, so that was all the information. Anybody have any questions? No? Okay, thank you for your patient hearing. And I okay, I'll be discussing the collinearity concept tomorrow, and the ambiguity we had regarding the size of mitochondrial chloroplast, that also I'll clear in the next class. Okay. Okay, that's it for today's session. We have a class at 8 o'clock. Okay, agronomy, herbicide classification. So be attend that, and okay, bye. I'm ending the meeting over here.